Okay, so what if I told you that one of our most powerful weapons against one of humanity's oldest diseases is actually a simple staining technique from over 100 years ago? It sounds kind of crazy, right? But it's true. This is the story of a stain that, honestly, helps save millions of lives.
And you might look at the date on this quote, 1998, and think, well, that's old news. But that's the scary part. This warning from the World Health Organization is actually more relevant today than ever. See, tuberculosis, it's not some dusty disease from the history books. It's a modern crisis that never really left.
And this is the human cost. 2 million people every single year. Think about that. That is a staggering number. And it really drives home just how critical it is to have a fast, cheap, and reliable way to diagnose this thing.
All right, so how do we even begin to fight a killer that's been with us for ages but is still so deadly? Well, the battle starts with one key thing: detection. You have to find the most infectious people as quickly as possible to stop the spread.
And this is where our story's hero comes onto the scene. You know, in a lot of the world, the first line of defense isn't some fancy, expensive DNA test. Nope, it's this simple, elegant procedure we're talking about today: direct smear microscopy. Why? Because it's cheap, it's fast, and it works. It's an absolutely essential tool in the fight for global health.
So you gotta wonder, right? What's so special about the tuberculosis bacterium? Why does it need this whole unique test just for itself? Well, it all comes down to its incredible defense system. Think of it like a waxy, nearly unbreakable suit of armor.
This special property has a name. It's called acid fastness. Basically, the bacteria's cell wall is just jam-packed with this waxy stuff called mycolic acids. The best way to think about it is like a biological raincoat. Once you get a dye inside that coat, that waxy layer just will not let it wash back out, especially not with acid.
And this is where the genius of the test comes in. Because the principle is so simple, it's brilliant. Here's what you do. You stain all the bacteria on a slide with a red dye. Then you wash it all with an acid. For almost every other kind of bacteria, that red dye just washes right out. But M. tuberculosis, that waxy armor, holds on to the red stain for dear life. It just refuses to let go. Okay, so you've got this cool scientific principle. But how do you turn that into a reliable, everyday test? Well, back in the 1880s, a couple of scientists, Franz Ziehl and Ziehl-Neelsen, figured it out. They developed this incredibly clever method specifically to take advantage of this whole acid-fast thing. Their method, which we still use today, is just this beautiful three-step process. First, you hit the slide with a powerful red dye called carbofuscin, and you add a little heat. That heat is key. It kind of melts the waxy armor just enough for the red dye to sneak inside. Step 2, you decolorize. You wash the slide with an acid-alcohol mix, and this strips the red dye from everything except our acid-fast bacteria. And then finally, the counterstain. You add a blue dye, which colors all the stuff that's now colorless. And voila! You've got this perfect visual contrast. And this isn't just some textbook theory, okay? This is a hands-on, precise procedure that happens every single day in labs all across the globe. And it's not something you can just wing. It takes a lot of skill and, really importantly, super strict safety rules. Because let's not forget that people doing this are handling samples that can be highly infectious. So, after all that careful staining and washing, the slide is finally ready. This is the moment of truth. Time to put it under the microscope and decode the results. And it all boils down to a really simple but really powerful color code. So imagine you're the technician. You're looking through the microscope, scanning the slide. What you're hoping not to see, but what you're looking for, is unmistakable. A positive result is these bright red little rod-shaped bacteria. We call them bacilli, and they're just glowing against this vast background of blue. It takes a really skilled eye, because sometimes you're just looking for one or two of them in a huge field. But here's a really important detail. It's not just a simple yes or no. The number of bacteria you see is a huge piece of the puzzle. That's why there's this grading scale recommended by the World Health Organization. It lets the lab report not just if the bacteria are there, but how many are there. You see, getting a 3+ report, well, that tells the doctor that the patient has a massive amount of bacteria and is extremely infectious. And that brings us to a really serious question. When you're dealing with millions of lives and the health of whole communities, what actually happens if you get the result wrong? Well, the stakes couldn't be higher. Think about a false positive. That means you tell a perfectly healthy person they have TB. You put them on months of powerful drugs that can have some really nasty side effects. But a false negative? Oh, that can be even worse.
Not only does the sick person not get treated, letting the disease get more severe, but they're also out there, completely unaware that they're spreading it to their family, their friends, their community.
So this leads to a pretty obvious question, right? In our modern world of high-tech medicine and DNA analysis, why on earth are we still relying so much on a technique that's like 140 years old?
Honestly, it all boils down to one word: practicality. Sure, those new molecular tests are amazing, maybe a little more sensitive, but they are also way more expensive and take a lot longer to get a result. Acid-fast microscopy, on the other hand, gives you a solid, reliable answer, and it does it fast and cheap. And that, right there, makes all the difference in the world. Literally. In so many countries, this simple test isn't just the best option, it's the only option. It is a fundamental pillar of global health equity. It means your ability to get diagnosed doesn't depend on how much money your local clinic has.
Which really leaves us with something to think about, doesn't it? In this age of DNA testing and incredible high-tech diagnostics, why does a 140-year-old staining technique remain a pillar of global public health? Maybe it's a powerful reminder that sometimes the best solution isn't the newest or the fanciest one. Sometimes it's the one that's simple, tough, and available to every single person who needs it.